Welcome to all TYBSC students. Myself, Lect Assistant Professor in Department of Zoology, GMD Arts, BW Commerce and Science College, Sinnar. Now today we will discuss about the cell biology chapter 3rd, structure of plasma membrane. In that, the first fluid mosaic model. The fluid mosaic model of plasma membrane was proposed by scientists Singer and Nicholson in 1972, and it is widely accepted as compared to other proposed models. The important features of this model is that the biological membranes are considered to be quasi-fluid, half-fluid, structure in which the lipids and integral proteins are arranged in a mosaic manner, and these models explain very well the properties of the cell membrane. The model assumes that there is a peripheral layer of phospholipid molecules in which peripheral and integral globular proteins are embedded to varying degrees. That is a fluid mosaic model of Singer and Nicholson, showing that carbohydrate, phospholipid bilayer, proteins, fatty acid tail, phosphate head. Now structure, how to explain it, that the content of the fluid mosaic model were worldwide accepted. Because of that, the certain immunological experiments, the attachment of the cell surface expose integral membrane proteins such as glycophorin, which lead to realization that membrane proteins are not fixed within the lipid layer but are free to move laterally like they are submerged in lipids. This observation inspired the name fluid mosaic model, coined by Singer and Nicholson. Because of that, the proteins and other carbohydrates which are submerged on the lipids layer, and therefore that is called as the fluid mosaic model. This model consider that lipid protein association to be hydrophobic, but the Daniel Dawson models assume hydrophilic bonding between lipids and proteins. The fluidity of the membrane is the result of hydrophobic interaction. The phospholipids and many intrinsic proteins are amphipathic molecules, that is both hydrophilic and hydrophobic group in same molecule. Proteins are contact with aqueous solvent on both sides of membrane. Generally, proteins are amphipathic, having both hydrophilic and hydrophobic groups. Their hydrophobic polar heads protrude from the surface of the membrane, while non-polar regions are embedded in interior of membrane. The integral proteins are capable of lateral diffusion, that is the transport across the membranes. The main function of plasma membrane is regulate the flow of material in and out of the cell, called transport. This transport of material is regulated by the size of pores present in plasma membrane. It is the semi-permeable membrane which allows the passage of solvent but not of solids. The plasma membrane, which permits one substance to pass through more easily than another, is called selective permeable. Now, types of transportation through plasma membrane: passive transport and active transport. In passive transport, there is also simple diffusion, facilitated diffusion, and active transport, primary active transport, and secondary active transport. Now, first, passive transport. Simple diffusion. The simple way for a solid to move from one side to membrane to other is simple diffusion. It is the unassisted net movement of solid from a region where its concentration is lower and concentration gradient disappear as diffusion occurs. There is no stereospecificity and the process is slow and ATP is not utilized. Now see the transfusion or diffusion of the uh, material through the plasma membrane, where there is a presence of the pores, and the pores which is formed by the protein channels, the protein molecules which form the pore channels in the plasma membrane, and they passes the gradient through the that channel. And see this, the higher concentration, simple diffusion showing the higher concentration to the lower concentration of the gradient. Now the structure of the plasma membrane showing that the arrangement, like as the double layer or bilayer of the lipids having the non-polar end and polar end with the attachment of the phosphate, and therefore that is called as the phospholipids, where there is also spread on the plasma membrane. There is a presence of the cholesterol in the form of the carbohydrate, then glycocalyx which is also the glucose containing the phospholipids, that called as the glycocalyx, where the proteins are also suspended on the surface of the plasma membrane, and that is called as the um, fluid suspended molecules. And where there is also two types of the protein, that is the integral protein and exterior proteins, or that is called as the peripheral proteins. The peripheral proteins which are also supported by the cytoskeleton, and the integral proteins which is also form the uh, channels to the ingredient, or form the pores of the plasma membrane. Now, in the type of diffusion, energy is not required and it takes place along the concentration gradient, for it differs from simple diffusion in some aspects. The carrier for transport of the metabolite across membrane, the stereospecific process, that is the only one isomer is transported. The roles of carrier protein is simply to facilitate the diffusion of polar or charged solute. Carriers are proteins with relatively low molecular weight, that is a 9 to 40 thousands, but they are highly selective. The metabolite binds the carrier proteins at the outer surface of the membrane to form carrier metabolite complex. The diffuses along the concentration gradient, that is from high concentration to low concentration organs. The metabolite is then released toward the inner surface of membrane is they are as there is a low concentration of metabolites on inner side. It is continue as long as concentration gradient is a download at the uh, at glucose in RBC, for example glucose in RBC. Now simple diffusion, see this higher concentration of the gradient which is passes toward the low concentration, that is the solute passes toward the low concentration through the plasma membrane pores or channels. Thank you very much. Now the next